morning. Today I'm presenting on what the news outlets have coined as the antibiotic apocalypse. So first, let's have some definitions out there. Does anyone know what a germ is by definition? I'll take any guesses. A germ. Okay, so a germ is an umbrella term for any microorganism that causes a disease. That could be a fungi, bacteria, a virus. A bacteria is a specific type of germ. It's a prokaryotic organism. And it's a domain, and there's lots of different types of bacteria, which means it can't all be treated by one antibiotic. There has to be multiple antibiotic cures. An antibiotic, which I just mentioned, is a drug that treats bacterial diseases by killing off the bacteria within your body. And a superbug is a bacteria that's adapted to its environment, whether it be through taking in a plasmid or an extra piece of DNA, or mutating its own DNA, so that it's no longer treatable by the same antibiotic as before. Yeah, those are the definitions. So the antibiotic apocalypse really is grounded in one concept, and it's that the more we use antibiotics, the higher bacterial resistance we'll have. So bacterial resistance is basically what I mentioned as superbugs. The more you use an antibiotic, the more superbugs will occur, and that means those superbugs won't be treated by the same bacteria drug as before, and we'll need a stronger drug to treat them. So to kind of represent this, I made a very, very simplified visual. Say this is a pool of bacteria, and remember, bacteria covers like every surface of everything in the world. So this is like super simplified. So let's say that all these green bacteria are like disease-causing agents, tuberculosis, let's say. We want to kill them all off because we have drugs to do so, and we don't want to get sick, of course. So we kill them off, we make them less visible to the environment, and the world is safer. We don't get sick from tuberculosis anymore. But if we overuse this drug, we kind of leave this space open for bacterial resistance to grow in. So let's say there's a red one, it's tuberculosis, with a piece of DNA inside of it that makes it no longer treatable by the same tuberculosis drug as before. As we kill off this tuberculosis grows in its place. We, of course, can make new drugs to treat this new antibiotic, new bacteria, but then we have the same issue where we overkill this bacteria, and we have a new resistance in this place that's, say, developed from a mutation in DNA replication. The only issue this time is we don't have a drug that's strong enough to treat the blue bacteria, and that's kind of the situation that's happening in the real world right now. We have a bacteria on our hands, a superbug, that we're not able to cure with modern science. I don't coined it the Callistan crisis in alliteration with the antibiotic apocalypse. So Callistan is a last resort drug among a whole pool of last resort drugs. It aims for a specific type of bacteria, and it's very good at killing it off. If you have no other, it's like a fail-safe basically. If no other drugs work, you turn to Callistan to kill off this type of bacteria. It's been overused by several countries in the field of agriculture. So China's farming pigs, obviously, to sell pork. To compensate for the very, very poor living conditions that they keep this pig in, so let's say they keep it in a dark, warm, muddy environment, which is perfect for bacteria to thrive in. They feed it antibiotics so that this bacteria is killed off, and they can make a really, really cheap hamburger and send it to the United States, and we could all eat McDonald's for $1. But what this done is that they've overused Callistan, and Callistan is a drug that you don't want to overuse, because like I said, it's last resort. So as you overuse it and the resistant strains become more prominent in the population, we now have, since it's last resort, we have bacteria that we can't cure with modern medicine. And that's exactly what's happening. There's a bacteria called MCR1 that we just don't have a cure for, and it's been projected to kill as many as more people than cancer by 2050, among other bacteria, superbugs. So it's a very huge issue that's facing society that's kind of been ignored. My paper is called New Antibiotic Kills Pathogens Without Detectable Resistance. I would simplify the title, but it's pretty much simple as it is. First, let's discuss culturable versus unculturable antibiotics. Culturable antibiotics are basically, culturable bacteria are bacteria that we can take from the environment, say from mud or soil, isolate it in a lab, and it will grow normally, it'll multiply, and then we can examine it and create antibiotics from it to cure that bacteria. Unfortunately, only 1% of all of the bacteria in the world is culturable. And we really didn't know the reason for this before, because 99% of the bacteria in the world couldn't multiply under lab conditions, like in a agar plate, all that. We wouldn't be able to multiply in those conditions, therefore it's kind of useless to a researcher or a scientist. We discovered recently, we still don't know all the details, but it's known as the Great Plate Count Anomaly, and what it might have to do with the environment that the bacteria is in. So say we have a bacteria in a deep trench. If we take it out of that trench, deprive it of all the growth factors in that environment, all the other microorganisms, all the other bacteria in that environment, isolate it on a single dish, it won't be able to multiply, it won't be able to grow, and therefore it'll be useless to us, and we can't really get antibiotics from it. Uncultured bacteria, which I mentioned, is 99% of bacteria. They can't survive under lab conditions normally. So what this paper talks about is the iChip. It's really like a revolutionary piece of technology that's really quite simple. It's like a plastic little disc this big with 172 little dishes in it, little diffusion chambers. You would put it in an environment, so mud, you would put it in water, you would put it in a trench, and it would diffuse everything except for one bacteria in each of its plates, while keeping, but since it's a permeable membrane, it's still within the environment of this bacteria, which means it won't die off, which means we basically have like a tiny little farm of these bacteria that we can take, examine, as it dies, take, examine. This is really revolutionary because now we have access to a whole huge part of the world, 99% of bacteria, and I'll remind you that bacteria is literally everywhere. So 99% of bacteria across the world is just a huge amount. We have access to that bacteria, which we could then synthesize into antibiotics, and that's really revolutionary. We have a huge pool of bacteria and antibiotics that we just look from. So one of the antibiotics that they've gotten from this method, from the iChip, is called Teixobactin. Teixobactin targets lipids 1, 2, and 3, and what those are is they're peptidoglycans, which create the cell wall of a bacteria. By targeting these, there's a few positives to it. One is that since we're not targeting the gene pathway, normal mutations like a misread nucleotide won't affect the functioning of the thing because it's not going after proteins. Teixobactin will still be able to kill off the cell wall of these peptidoglycans. Another benefit of it is that it has 3 targets, lipids 1, 2, and 3. The leader in cell wall attacks has been vancomycin, and it's only targeted lipid 2 in the past. So if lipid 2, for whatever reason, wasn't available to that drug, it wouldn't be able to kill off that bacteria. Whereas Teixobactin has 3 targets, so each of them is a failsafe for the other. In our first little figure here, it basically says what I just said. When you have a dosage of 2 Teixobactin per lipid, there's no lipid 1, there's no lipid 2, and there's no lipid 3, as well as some other peptidoglycans. That just shows that it has multiple targets. And over in this figure, the dark bars are the Teixobactin levels, and the white bars are the leader in that type of drug. So in DNA targeting drugs, ciprofloxacin has a very low level of DNA, and that makes sense. It targets the DNA of a drug, of a bacteria. Teixobactin still has a huge amount because it doesn't go after that part of the bacteria. RNA, same with this one, very low levels of RNA. Teixobactin doesn't target it, so it's still high up there. But as we can see with peptidoglycans, the level is very low. There's a 0% incorporation rate, which means very little peptidoglycans are being synthesized when you insert Teixobactin into it. It's about on par with vancomycin, which is the other leader in lipid targeting antibiotics. But a benefit that Teixobactin has over vancomycin is it's way more bactericidal, which basically just means it kills off a lot more bacteria a lot more efficiently. So in this experiment, they did serial passage of bacteria, which means they periodically inserted bacteria into a plate and saw how Teixobactin and vancomycin dealt with it. Vancomycin brought it down, but eventually when the levels of bacteria went back up. By the way, log colony forming unit basically just means the amount of bacteria. So the amount of bacteria essentially went back up because of the serial passage. They kept inserting bacteria, and I guess vancomycin just couldn't handle it. But Teixobactin level consistently went down. It killed off bacteria a lot more efficiently than vancomycin did. Now, it has a very low dose dependency. So basically, in this figure, we see the percentage of survival of mice, let's say. Normally, the infection control, mice would just die, almost 0% survival rate from the bacteria that they put inside of it. But when you insert vancomycin, you need 5 milligrams per kilogram of mice, to have a 100% survival rate. Teixobactin only needed 0.5 milligrams. So by having a very, very low dose dependence, it's super efficient. We can ship it off in much bigger quantities because you only need so much to cure a certain amount of bacteria. It's just really good economically wise. Resistance. So fold change in minimum inhibitory content essentially just means the fold change in the amount of antibiotic that we need to kill a bacteria. In this one, we see ofloxacin, which doesn't target the cell wall, which is why it has so much fold change. It goes up over the course of 25 days. The fold change of this exact kind of keyhole for this bacteria goes up a lot. It's still treatable by ofloxacin, hasn't changed that drastically, but it goes up a lot compared to teixobactin, which had virtually no fold change. This is, like I said before, because of the targeting of the cell wall. It won't really react to any fold change of the gene pathway. Vancomycin is a lot like teixobactin. It didn't have a resistant strain discovered from it for 30 whole years. So upon its discovery, it took 30 years for them to find a strain of bacteria that couldn't be treated by vancomycin, kind of in the range that vancomycin treats. So hopefully that means teixobactin has a very long lifespan ahead of it. So what's the future of this paper? Teixobactin, as great as a drug as it is, isn't a solution to the colistin crisis because it just simply doesn't target the same bacteria that colistin once did. So it doesn't solve that crisis individually, but why I mention it because it shows the strength of the antibiotics that the iChip is discovering. The iChip discovered teixobactin within its first year of creation, and teixobactin is like really an amazingly impressive drug. It's the best in its field since its discovery. But there are many, many more antibiotics to test. Like I said, 99% of the entire world's bacteria. We still have to test them and find antibiotics for them. This includes bacteria from the very deepest seas, which we haven't even fully discovered yet, or even bacteria on other planets. Like we've heard of bacteria growing on foreign planets. We could harvest that using the iChip, keep them in safe conditions within their environment, and then turn them into antibiotics. It's a little far off, but maybe it'll happen one day. So yeah, hopefully the iChip, through its the depth of its discoveries, we could find a cure to the colistin crisis and a cure to any future crises we may run into. But ethics. So we can't really rely on the iChip as a duo ex machina. We can't rely on it as an end-all be-all solution to any problems we may have with bacteria in the future, because that's just really not a safe thing to do. We don't know the extent of the iChip, so we can't rely on it. What we should do is have stricter regulations on factories and farms. So even though it'll make your burgers or your food a little more expensive, it's really important so that we don't have any other crises on our hands, like the colistin issue. Yeah, so we have to really follow a strict line of ethics and learn from the situation that we're currently in. Thank you. Journal team. Yes.
What other planets have bacteria?
Yeah, sorry. I think did they say Mars had water or something? Yeah, so we can check the water in Mars. Or any other moons we discovered in the future. I don't know. I'm not an astronomy person. Anyone else? All right. Oh, yes.
So this drug was isolated from bacteria that previously had been unidentified.
Yeah. Well, it's been identified, but we haven't been able to grow it in lab conditions because it was unculturable.
Okay. So this is a compound then that the bacteria makes on its own and secretes.
Teixobactin?
Yeah.
Yeah, I guess. It could have been synthesized, some antibiotics are synthesized from the bacteria it's discovered, specific to that bacteria.
Okay.
But I think in this case, I think you're right, it was done like that.
They isolated it from something that the bacteria produced.
Yeah. Anyone else? Yeah.
Do you have any idea what fractions of 99% in culture by normal means the chip allows us to get?
I think virtually all of it. The only foreseeable issue I could see is if keeping the bacteria in its environmental condition isn't enough. But I don't really see that as an issue. I can't see a situation where that would happen. So I think it really gives us access to all of it. But of course, that isn't immediate access because it takes a really, really long time to test and put drugs through trials. So it's not like we're going to get 1 billion antibiotics in the next year. It still takes a really long time for them. Teixobactin is still in human trials. It was discovered, I think, 2 years ago. So it's taken a while. Yes?
So the antibiotic has an impact on that particular bacteria. What other impacts might it have? You talked about the ethical, but I'm wondering along the lines of the biological impact that antibiotic might have.
Of teixobactin?
Yeah.
I'm not sure exactly what you mean.
So are there any other residual impacts? So you're trying a direct line connection between the bacteria and
Yeah, so the way bacteria works is, there's, like I said, the bacteria is a domain of species, so there's a lot of different types of bacteria. So Teixobactin targets, I think, gram-positive, targets a branch of gram-positive bacteria, so again, it's like a branch of bacteria. So it's not like it targets only one type of bacteria, but it targets kind of a smaller pool of them. But that's the case with many antibiotics, with many antibiotics as well. So yeah, we don't really expect it to cover any more than what it's like been able to target. I couldn't say exactly why it targets that specific branch of bacteria because I don't really know enough about gram-positive versus gram-negative bacteria. But yeah, it targets a specific pool.
Are you asking whether there would be other harmful effects to using this?
is there the potential for that, or is there anything known yet?
Well, since it's actually targeting the lipids in the cell membrane of the bacteria, so it shouldn't be affecting like our cells, if that's what you're asking. I don't know.
Any cells, I
Or
guess,
like helping other good bacteria maybe, or
or good bacteria.
yeah.
that
Well, with any
Yeah.
antibiotic, it does, but that's why when you take antibiotics, you eat yogurt so that you replace some of those good bacteria.
Yeah, it's definitely an issue with like bacteria in general. It kills off good bacteria as well. I neglected to mention it, but there are also good bacteria in
Yeah.
the world. So yeah, that could definitely be a foreseeable issue. Yeah. Or
Is that what you're asking?
Yeah, I was thinking more about the microbiome and how expansive the impact
Yeah.
What effect would like repopulation? So you talked about the problem is that when you kill off a lot of bacteria, you allow for resistance rates to start reproducing and fill that empty space.
Yeah.
But what happens like when you eat yogurt then and like kind of replace the bacteria that were already there? Would that lessen the effect?
Well, yeah, basically, it replaces it replaces like the bacteria that you just killed off, the good bacteria, with more good bacteria. So that kind of like that airspace is taken up by things that won't harm you. Eat pig's yogurt. Well, unfortunately, the good bacteria, I guess, they're a part of the immune system, I think. So the immune system isn't really strong enough to face like diseases like MCR1, which defeated colistin, unfortunately. So yogurt wouldn't be a solution. But I like your line of thought. Anyone else?